Welcome to the last video in the Immune Disorders and Vaccination unit. In this video, we're going to talk about the different types of vaccines that are used in vaccinations. I'm going to give you examples of some of those. We're also going to look at vaccines as a public health measure and why they are so beneficial. Let's go ahead and take a look. The first vaccine we'll talk about are the live attenuated vaccines. These tend to be one of the more common, and they tend to also most closely mimic a natural infection. So they tend to be some of the best vaccines at actually activating an adaptive immune response. These vaccines are going to be living cells. So for example, if this here is the pathogen, and remember we've got proteins on the surface of the pathogen, those are good targets for antibodies because they're floating out there. We've got some genetic code inside. What the vaccine would be is that that genetic code picks up a bunch of mutations. And so we have the genetic code, but it gets mutated in a number of spots, which changes the pathogen itself so that it's no longer a pathogen. So these are non-pathogenic, but it hasn't changed it so much that it doesn't look like the pathogen. So you can see the proteins out here on the surface still look similar to what they had been, so that they're still going to mount an immune response to something that looks like the pathogen, but it's not actually pathogenic. The reason these are advantageous is because since they're alive, they actually do replicate inside the body. This means they do set off an immune response. They are a foreign molecule that's invading and can grow and divide, but they're really not pathogens, and so the immune system wins and gets rid of the microbe itself. The disadvantages to this is that because it is actually a replicating microbe, you typically can't use these in immunocompromised individuals because their immune system isn't as strong, and so that replicating microbe could become a pathogen in those small populations. There's also a very small possibility of reversion back to the pathogenic version. So if you didn't have enough mutations here in the genome, you can sometimes get what's called reversion mutations, where they mutate back and it becomes pathogenic. There's actually an outbreak of polio in India a year ago. That strain of polio actually looked very similar to the vaccine strain, and there was worry that maybe it had been a reversion. The other problem with live attenuated vaccines is because they are living, you do need to maintain them as live cells. So you have to be growing these things, you have to store them in cold temperatures so that the cells stay alive, because they have to be alive when they get injected into the body. Examples of these are the MMR vaccine, which is the measles, mumps, and rubella. TB also has a vaccine, although it's not the best vaccine in the world, but there is a vaccine for TB that is also a live attenuated version. The next common type of vaccines are inactivated vaccines. These are vaccines where you're using the pathogen, but the pathogen has been inactivated, so it's dead. So for example, if you had your pathogen like before, you would take this pathogen and you would expose it to something like high heat or various chemicals, and that's going to kill the pathogen itself. And so it still looks like the pathogen, which means you can still mount an antibody response to the proteins on the surface or the proteins of the pathogen itself, but it can't replicate, it can't grow because it's not actually alive. And so one advantage of inactivated vaccines is you can use them in someone who is immunocompromised because the pathogen can't reproduce, so they're a little bit safer that way. There's no way they can reconvert to a pathogen because they're dead. The disadvantage is because they're not actually growing, the body doesn't treat them quite the same as it would a living microbial pathogen. And so these are typically the vaccines that need a booster. So something that's going to strengthen the immune response, because often you only get a weak immune response and then you have to give it again and you get a little bit of a better one, and you give it again and you get a better one. So for something like hepatitis A or rabies, these can be used as inactivated vaccines, but you often need a booster dose. And the problem with that is sometimes patients don't always follow up for those additional doses. Two other types of vaccines that are becoming more and more common are subunit vaccines and toxoid vaccines. These are both very similar. The difference is just the subunit vaccines use only a fragment of the pathogen, so some particular protein, whereas the toxoid vaccines are using a toxin, so the toxin protein itself. The difference there is that if it's a toxoid vaccine, you have to inactivate it first, or else you would be giving someone the actual toxin. So what this looks like is Again, here's our pathogen. But now, instead of actually using a cell at all, so we're not using a live cell, we're not using a dead cell, instead we're using genetic engineering to just make part of the pathogen itself. And so a good example of a subunit would be to take these proteins on the surface that maybe were the spike proteins from a virus and use genetic engineering to make cells that are just going to make a bunch of the spike proteins that you could then purify and inject into the body as the vaccine itself. You can do the same thing with a toxin. So if this pathogen made a toxin protein, you could genetically modify and use that toxin. But remember, if you were going to do that, you would then have to heat it up so that you destroy the structure of the toxin, mainly so that you don't inject the functional toxin and actually cause disease. Some of the common ones we see with this are the DTaP, which is the pertussis, tetanus, and diphtheria toxin. Those are a subunit of pertussis, and then the toxoid vaccines for tetanus and diphtheria. And HPV is also a subunit vaccine. The key with these is they often have to be given along with what's called an adjuvant, and that's just something that's going to help enhance the general immune response. Some common adjuvants are things like aluminum or calcium salts, and it's really because you're not giving a cell itself, which is what the immune system is used to responding to. And so adjuvants just help boost the immune response so you get a better memory response forming. Some of the newer vaccines are recombinant vaccines or DNA vaccines, where instead of injecting the protein itself, you're actually using the DNA level. So you can actually take the DNA for those proteins you might want to use from a pathogen and just use the DNA itself. One way they can do this is put the DNA onto gold particles and then actually just shoot it into muscle with what they call a gene gun, shown here. And that DNA gets into our cells are able to transcribe and translate it into protein. So here you can see the DNA gets into our cells are going to transcribe and translate those proteins. They get expressed out on the MHC1. That MHC1 then activates T cells and induces the adaptive immune system response. We're also now using recombinant vaccines where we take the DNA for certain pathogens and put it inside a different bacterial cell or a different viral particle that are not pathogens. And then we let the bacteria or the virus deliver that DNA for us. These new DNA vaccines and recombinant vaccines are really what's coming down the pipeline in new vaccine development. So no matter the type of vaccine given, vaccines are really one of the biggest public health victories in all of human history. They've dramatically reduced morbidity and mortality in all different age groups, and it's really because vaccines allow prevention of the disease. As opposed to things like antibiotics or antivirals that are treating the disease, vaccines stop anyone from getting it. This difference means if you have a disease and you're taking an antibiotic for it, you still could be passing that disease to others, which means it's spreading through the population. As opposed to vaccines, which prevent you from ever getting the disease, if you don't get it, you can't spread it to someone else. And so when we look at public health in the next week, we're really going to look at how do we keep public safe from diseases, and vaccines are one of the best ways to do this. And you can see this list here of the 10 greatest public health achievements in the 20th century. Right at the top of it are our vaccinations. Keeping on a vaccination schedule really prevents about 48,000 deaths and about 20 million cases of disease every year, and saves society as a whole about $69 billion. And there's a lot of money spent to treat diseases that if you can prevent them in the first place, you don't actually need to spend. Vaccines are a great public health measure because they work. This is a comparison of the number of cases in the United States of certain diseases based in 1900s compared to 2010. Now, the 2010 data is a little bit old, it's about 10 years old, but it still gives you a really good sense. So for example, this big yellow circle is measles, where back in the 1900s, we were having over 530,000 cases every year. In 2010, we were down to this little tiny circle, which was 61 cases. Now, unfortunately, with some of the vaccine hesitancy that is more prevalent in society these days, that little measles circle has started to grow, and we're going to talk about that a little bit in your vaccine assignment. If you look at some of the other numbers, you'll see a number of them have now dropped to 0. So you can see smallpox, we have no cases. Remember, there's no cases anywhere in the world. A diphtheria, no cases in the United States. Polio, no cases in the United States compared to 16,000 back then. And others that are down in the single digits. So 6 cases of rubella, 8 cases of tetanus compared to 47,000 cases of rubella. And so a huge difference in the number of people who are getting these diseases and why it's important to continue to vaccinate for them. Vaccination is important because it protects you individually from getting a disease, but it's also an important public health measure because it can protect populations from being able to spread the disease throughout them. This concept is called herd immunity. Herd immunity works like this. All the blue people here are people who are not immunized, but they're healthy people. And you notice a couple of people in the crowd get a disease. That's your red folks. Those infected people then spread it to everyone around them. And those people spread it to people around them until you have a population where lots and lots of people are now sick and contagious. This is what we're seeing a little bit of with COVID-19 because no one in the population had immunity to this disease as it was a novel pathogen and we had never seen it in humans before. If you do have some people who are immune to the disease, either because they've gotten vaccinated or they've already had the disease at some point in their life, they're marked here with the yellow folks. But if you only have a couple of them, then they're not enough to present the whole population. So we've got our couple of infected individuals, and again, notice they're coming into lots of contact with people who are not protected. And so we still had a couple of those folks who didn't get sick, but the majority of the population still ended up with the disease. Herd immunity occurs when most of the population has been vaccinated. So you can see most of the population now is those yellow protected individuals because they've been immunized. We had a couple in the population who were not, who have gotten sick, but now when they come into contact with people, mostly who they're coming into contact with are vaccinated people. And because they're going to mount an adaptive immune response and not actually get sick, they won't be contagious. And so this stops the spread of disease in populations, and it allows us to protect those individuals in the crowd who were not vaccinated and who were not immunized. This is important because there are some people who are immunocompromised who are not able to get vaccines. And there are also some people who are, say, infants that are not old enough for vaccines yet. And having herd immunity helps protect those individuals from exposure to the disease. The percentage of the population that's required to be vaccinated to achieve herd immunity depends on the infectivity of the pathogen, so how likely is it to spread from person to person. For example, measles is one of the most infectious pathogens we know, and to have herd immunity against measles, you really have to have about 93 to 95% of the population vaccinated to protect those individuals who can't get the vaccine. Now, I'm sure you've all heard the term anti-vaxxer. In the scientific community or in the medical community, we really use the term vaccine hesitant. That's because these are going to be people you might be interacting with on a professional level that you're trying to convince to vaccinate their children. Anti-vaxxers sound a bit derogatory. You want to actually be educational and helpful while interacting with people. You're going to do an assignment based on vaccine hesitancy where I'm going to have you look at some data. So I'm not going to go into that right now. There'll actually be another video for you to watch next week after you've done the assignment. But I did just want to mention that all drugs have some sort of side effect. And so while vaccines don't necessarily cause the type of concerns that some people have, they do have side effects and there are differences in their effectiveness. So for one example, most vaccines are going to give you an inflammatory response. Remember, you are trying to set off the adaptive immune system. Before you get to the adaptive immune system, you have to go through the innate immune system and that inflammation response. What this means is you might get some swelling and some discomfort at the injection site. That's the inflammatory process happening in response to the foreign antigen. You might also feel a little bit ill, so you might run a fever. Remember, that's the innate immune response. Those are good things. It means that your body is responding to the vaccine the way you want it to, and it's going to mount a good adaptive response so you're protected later. What vaccines don't do is give you the actual disease. So just because you might be feeling a little bit ill after you get the influenza shot doesn't mean you have full-blown influenza. There's also the possibility for allergic responses. Just like with any drugs, there's a certain portion of the population that is going to be allergic to some of the things in vaccines. So for example, some vaccines, like the influenza vaccine, are prepared by growing the virus in eggs. If a patient has an egg allergy, they can't get that type of influenza vaccine. We now make cell-based influenza vaccines for those types of individuals. And then the third thing is there is a difference in effectiveness. So based on your genetics, you have a slightly different immune system than anyone else around you. It's why some people seem like they get diseases or infections more often than others. Some people have strong immune systems and some it might be a little bit weaker. So not everyone has an identical response to the vaccine. And the other key is timing. So if you remember, that primary response takes a while to induce your adaptive immune response, about 10 days or so. This means that if you get, say, the influenza vaccine, and the very next day you're exposed to someone with the flu, that vaccine isn't protecting you yet. So there's a good chance you might get the flu. When this happens, that's when people say things like, oh, I got the vaccine and it gave me the flu, or I got the vaccine and it didn't do anything. That's because you're not immediately protected. The second they inject that vaccine in, you still need to go through all of the immune system processes that we learned when we talked about the immune system to mount that primary response.